Yeah, the recording has started. So let's break this bacteria slide. So bacteria are tiny living things, microorganisms that are how many cells in the bacteria? How many cells in the bacteria?
9.
9. Where the 9 came from?
Just guessed.
This is your lucky number, right? The 9 is the luckiest number, yours, I guess. So this is the single-celled organism, only one cell in the bacteria, okay? Single-celled organism and belong to group called the prokaryotes. They belong to the prokaryotes. What was the meaning of the prokaryote? Do you remember?
Prokaryotes. I can forget the meaning.
What were the prokaryotes? What are the prokaryotes? Prokaryotes are organisms without the nucleus. They are organisms without the nucleus, no nucleus in the prokaryotes, okay? So we call them prokaryotes. So, good. So bacteria are tiny living organisms or tiny living microorganisms that are single-celled and belong to the group that's called prokaryotes, okay? So prokaryotes that we call prokaryotes. So bacterial reproduction, how the reproduction of the bacteria is happening, we call them binary fission. Okay, this means they copy themselves by simply splitting into the two. So binary fission means splitting into two, splitting into two. Just they split into two, so they copy themselves by splitting into two. Okay, what are the steps? First, the single circular DNA is copied, so they replicate single chromosome. So DNA will be copied, one DNA will be two before the division. Okay, the cell is going to grow because they want to make two cells from one cell. That's why the cell should grow. Okay, and after that, the end stage, it splits into two cells that are genetically identical. From one cell, they're going to get two cells which are genetically identical. Okay, genetically identical. First, it's growing in size. The growing is happening. Their DNA is twice increasing. That means their chromosome is twice increasing. Okay, so they replicate the single chromosomes. Okay, good. Any questions so far? Example, for example, like they are like a photocopier making two identical copies of one paper. Okay, so you have a one paper, you want to copy it, so you're getting two copies on one paper. That's like a photocopying. Okay, why is this useful for the bacteria? Bacteria can reproduce very fast. Some can double every 20 minutes. Imagine, so one bacteria can be two bacteria in 20 minutes. It's very fast. They reproduce very fast. That's why they are um, and their reproduction is very fast. That's why the bacteria are more, microorganisms are more compared to the animals or humans. Okay, so what's the classification of the bacteria? First of all, let's learn the bacterial classification. So bacterial classification, they are single-celled organisms. They have only one cell, not 9, okay? They live and survive as one cell, only one cell. And only prokaryotes, they call prokaryotes, which means they have no nucleus, okay? They don't have the nucleus, only prokaryotes around. So prokaryotes means no nuclei, no nucleus, and there is no membrane bound organelles. There is no membrane bound organelles. For example, the mitochondrion or rough endoplasmic reticulum, they are absent, okay? What are the examples? What are the examples to the bacteria? For example, do you know any examples?
Bacterias. Uh, I don't know names
E.
because
coli.
I've learned them, but they're like very complicated names.
Yes, E. coli, for example. E. coli are in our intestines, okay? The intestines of the human body, they are inside of the intestines, or I can say the streptococcus. What they cause, streptococcus, they cause the strep throat, can cause the sore throat, okay? You take the amoxiclav after that. This is the antibiotics. Or they can be lactobacillus. Lactobacillus. Have you heard lactobacillus?
No.
Lactobacteria. Use it in the yogurt production. It's inside of the intestine as well to break down the lactose in the milk, to break down the lactose. As you grow up, as you age from the baby to the old age, the babies have more lactobacillus because they always consume the milk. Okay, so lactobacilli is going to break down that lactose, and as you age, as you grow up, you will have the lactose intolerance. So you will not tolerate the lactose containing products, for example, including the milk as well. Okay, so good. What's inside of the bacteria? Let's learn the structures of the bacteria. First of all, the capsule. Capsule. So what's capsule? Why we need the capsule? This is the extra outer coating for the protection. This is extra outer coating for the protection, okay? Like a raincoat of the cell. Like a raincoat of the cell, okay? This is called capsule. This is extra outer coating for the protection only purposes. And then below the capsule, you see this is the capsule. And below the capsule, we have the cell wall. What's the function of the cell wall? It gives the shape and the support. It's like a skeleton giving the shape and the support to the cell, okay? So what was the cell wall of bacteria?
Cell wall of a bacteria.
Yes, what was the name of the cell wall of bacteria?
Peptidoglycan.
Yeah, good job, good job, good job. Peptidoglycan, of course, peptidoglycan, okay? So the cell wall is made of peptidoglycan. It's like a skeleton, okay? What about the cell wall of fungi? What was the cell wall of the fungi?
Chitin.
Chitin.
Yes, exactly. That was chitin. Okay, chitin. What was the cell wall of the plant?
Cellulose.
That was cellulose. Exactly. The cellulose is a cell wall of plants. Good job. So plasma membrane. After that, we have the plasma membrane. This is just the membrane of the cell. What kind of membrane? This is cell membrane, okay? So it controls what enters and what exits. The function of the cell wall, it's like a gatekeeper. Gatekeeper. It's controlling what's going inside and what's going outside. So cytoplasm. Cytoplasm. Why we need the cytoplasm? It's a jelly-like fluid inside of the cell, right? This is the jelly-like fluid inside of the cell. It's like a soap where everything is floating, all the organelles are floating. Okay, ribosomes. Then ribosomes. Why we need the ribosomes?
It synthesizes protein.
Yes, so that good job. They are synthesizing the proteins. They are making the proteins. Okay, they are making the proteins. So why we need the proteins? They're essential for the cell survival, right? They're essential for the cell survival. Like they are like a factory machine which producing the proteins. Okay. So what's nucleoid? It is
Part of the bacteria.
nucleoid. Nucleoid. It is a DNA actually. Okay, that contains the genetic material, and they are circular. They are circular. They are the DNA of the bacteria is circular. Okay. They're circular, they're like blueprints or instruction books of the cell. Every instruction is written here, everything regarding the bacteria, okay? So what is plasmid then? What's plasmid? What is plasmid? Plasmids are extra small circular DNAs. Okay, plasmids are extra small, they're circular DNAs. They're circular DNAs. What are they going to do? They're going to have antibiotic resistance genes. You know what's antibiotic resistance?
To defend from antibiotics.
This is
They have
useful genes.
they're useful genes, of course, for the antibiotic resistance. For example, have you ever heard when you give a patient the antibiotic, it's not working?
No.
You're giving the antibiotic to the patient and it's not working because of the antibiotic resistance. So the bacteria contains the genes which make it resistant to antibiotic. Okay? Устойчивость, antibiyotik устойчивость ileri, antibiyotik çiğdemlilik. So they contains those genes in the plasmids. Okay? Good. What's pili? So what's pili? Pili, they are hair-like structures. They have hair-like structures to stick to surfaces. For example, E. coli bacteria is going to stick on the surfaces of the intestine, okay, and also to the urinary system. You know what's urinary system, right? Мочевыводящая система. Or exchange the DNA, or they exchange the plasmids. They exchange the plasmids with those pili. We call them sex pili. Okay, they exchange the information from one bacteria to another with these pili. Okay, good. It's like a USB connection. One bacteria is going to connect to each other and share their information. It's like a USB cable connection. Okay. Good. So what's flagello? What's flagella of the bacteria? Why we need the flagello? What do you think? Why we need this flagella? Just make your brain work, guys. Just
Bacterial flagellum
yes,
to move.
yes, tail-like part. It's a tail-like part. They use it for the movement. They're like a motor of the cell. Okay, so bacteria also moves with flagellum. Okay, don't forget. Yeah, it's for movement. So, guys, any questions regarding this?
No questions.
Okay, good. Let's check our knowledge a little bit regarding this. So what type of the cell is bacterium? Jahangir?
Oh, type of cell of bacterium.
Mm-hmm. Eukaryotic or prokaryotic?
Prokaryotes.
What's the meaning of prokaryotic?
Non non-membrane.
No
No,
nucleus. Without nucleus.
no
Okay, without.
nucleus.
Okay, good. So that which structure helps bacteria to stick to surfaces or other cells?
I don't know.
Flagellum, right?
To move?
I said to stick to surfaces or other cells.
Isn't flagellum to move?
No. Yes. Flagella for the movement. I am asking you for sticking to some surfaces.
I don't know.
Okay, Mahbuba.
I don't know too. Like
It's called
ribosomes.
sticking to the surfaces, Jahangir.
Really?
Which structure of the bacteria helps them to stick to the surfaces or to other cells? Guys, are you serious? I just talked about it. Are you concentrating in the class? They are pili. Pili is used for sticking to the surfaces. It's like a USB cable and changing for the information from one bacteria to another. What they're going to change with another bacteria?
Information.
Stored in the plasmid, yeah? Information stored in the plasmid. Okay, good. What is plasmid, Saadat? Mahbuba?
This is a piece of DNA.
Circular DNA piece. Good.
Exactly.
Okay. Good. How many cells do bacteria have, Jahangir?
One.
One.
Okay. What's the name of the process bacteria use to reproduce?
Mitosis.
Yes, mitosis, especially the binary fission. We call it binary fission. Okay, good. Okay. Good. What do ribosomes do inside the bacterial cell, Mahbuba?
Ribosomes.
Yes, what do those ribosomes are going to do inside of the bacteria?
They like use it for proteins.
Mm-hmm. Mm-hmm. To synthesize the proteins. They're used to synthesize the proteins, right? That's good.
Yeah.
Good. Any questions?
No
We
questions.
have talked about it as well, right? Bacterial DNA, circular chromosome, we talked about it. This is, let's start from this slide. Okay. So, how bacteria is going to share its DNA? Okay, when bacteria is going to share, how the bacteria is actually going to survive by sharing its DNA. This is called genetic recombination. They don't just divide and copy themselves. Bacteria don't just divide and copy themselves, okay? They also exchange their genetic information, which is circular chromosome, with each other in three ways, which we call the horizontal gene transfer, okay? We call them horizontal gene transfer. So they're going to exchange their information in three ways, okay? Good. The first process is called transformation. Okay, the second is called conjugation. The third is called transduction. Okay, let's learn each of them. First of all, transformation. What's transformation? So transformation means a bacteria picks up the naked DNA fragments from the dead bacteria around it. That DNA enters and becomes part of its genome. Okay, for example, here, this process is called transformation. Okay, environment to bacteria. What's the meaning of it? Bacteria, this is bacteria, it picking up the DNA, okay, this is naked DNA fragment, okay, is getting, maybe this DNA fragment is from dead bacteria, okay, from the dead bacteria around it. This bacterial information, genetic information is inserting, getting inside to one another bacteria. That DNA is going to enter and becoming a part of this genome, the part of this DNA. It's getting inside and sticking to another bacteria's DNA, okay? The DNA is floating in the environment, first of all, and then it's becoming inside. What's the example to it? The example for that, a bacteria becomes the antibiotic resistance after picking up resistant gene from its surrounding. For example, these bacteria contains the antibiotic resistance, right? This bacteria containing the antibiotic resistance. This antibiotic resistance gene is now entering into another bacteria. Now this bacteria is also resistant to antibiotic now. This is the first way. This is called the transformation. This is one of the ways of bacteria sharing its genetic information to another bacteria. So what about next? The next is conjugation. What's conjugation process? Bacteria to bacteria through the direct contact. It's written here, but how it works, but how it's going to work. Two bacteria physically connect using a structure called the pilus, okay? So this structure is called the pili. We learned the pili, right? Why we need the pili?
To
stick.
stick.
To stick to another
Cells.
bacteria, right? To
Surfaces.
stick to another bacteria,
Yeah.
to stick to another bacteria and exchanging the genes. It's like a USB, exchanging their information. So two bacteria coming together. This is the first bacteria. This is second bacteria and first bacteria. This is the pilus. What they're going to do, they're going to make a bridge. This is called the bridge between two bacteria, okay? They connected now. They connected to each other. So what's going to happen then? One bacterium, this is the plasmid, right? This is the circular DNA. This is the plasmid for exchanging the information. It contains what? It contains the antibiotic resistance gene. It contains the antibiotic resistance information. So this information is, I'm going to transfer this information to another bacteria. I want to, this bacteria contains the superpower. This is, for example, this bacteria is resistant to amoxicillin or tetracycline, okay, because of this plasmid, because of the resistance gene. And I'm going to transfer this to another bacteria. Bacteria work like that. They always support each other. If one bacteria has antibiotic resistance, they're always going to pass it to another bacteria. This is called the conjugation. So the direct contact with pili. And that information is going to another bacteria, okay? Another bacteria. And then you see now both bacteria have the plasmids. This process is called what? Conjugation. One bacterium transferring its plasmid to another bacteria. Key idea here, they touch each other directly. Do not confuse. This was transformation was from the dead bacteria, from the environment, the naked DNA coming to the bacteria, okay? But here conjugation, they have the direct contact with pili, sex pili we call them, because they're exchanging their information. A resistance bacteria, this is the resistant bacteria, can share the resistant plasmid with a neighbor bacteria, making both resistant at the end. They are both resistant now, okay? Both resistant now, both resistant now, okay? What about transduction? Transduction, this is the bacteriophage. So here you can see the bacteriophage. This is called bacteriophage. You know what's bacteriophage?
No.
Bacteriophage. So bacteria means bacteriophage means the virus. That means virus affecting the bacteria. Virus affecting the bacteria, okay? Okay, what's going to happen here? This virus, this bacteriophage is going to do what? This bacteriophage accidentally picks up some bacterial DNA when affecting one bacterium. Okay, that information, that DNA in the bacteriophage came from actually another bacteria. They picked up from one bacteria its genome, its DNA, and now it came to another cell. This is another cell, another bacterium, and inserting the previous bacterial genome here. Okay, then it injecting the DNA. This is called injecting the DNA, injecting the DNA into another bacterium during the next infection. Key idea here, DNA is moved by a virus phage. This is the DNA. This is the DNA of bacteria actually, and this DNA of bacteria traveled. It's like a plane for them. It's like a plane for the bacteria, and inside is bacterial DNA. Okay, this bacterial DNA is transferring through the virus and getting to another bacteria. A virus transfers genes to another bacteria. Okay, or DNA is moved by a virus, I can say. So a virus transferring the toxic genes from one bacterium to another, making it more harmful, for example. Okay, good. So you know the transformation now. Transformation, this is from the environment, right? From the environment, the DNA is transforming to another bacteria. Now you know the conjugation process. The conjugation means what? Two and not two bacteria are going to join together and transfer their plasmids. And transduction means the virus, which we call the bacteriophage, okay, transfers the virulence genes to another bacteria. Good. Any questions? I hope you understood the
question.
process.
Yes.
Okay, good. Here, the illustration of the conjugation. Conjugation means two bacterial cells are going to share their information, right? The conjugation means the bacteria replicates its DNA and donates some of it to another bacterium through the bridge called the pilus. You see, this is the information, okay? This information, antibiotic resistance information, they're making the bridge with what? With what they are making the bridge?
With pills.
Yes, with sex pilus,
Pill.
and then pilus, yes, and then the information regarding the antibiotic resistance is going to another bacterium. Now both of them have the antibiotic resistance gene. So we talked about the transformation, right? We talked about the transformation from the outer source. From the outer source, what's going to happen? The naked DNAs from the dead cells are going to enter to the bacteria accidentally. And after that, what's going to happen? And after that, this bacteria also will contain the genes from the previous cell. So transformation is when the bacterium when the bacterium is going to absorb the naked DNA from its surroundings, usually from the dead bacteria always, okay? And adds its own DNA. Okay, and adds it to its own DNA. It's adding this information to its own DNA. The red one was its own DNA. Now the bluish ones came from the environment. Now they are adding to themselves. Okay. Okay, for example, here, naked DNA from the environment, from the dead bacterial cell, is released into the environment. Now it's in the environment, okay? This DNA may contain the useful genes, for example, the gene, antibiotic resistant gene, okay? For example, that can be cow's gene. Have you ever heard about the biomedical engineering? It's very popular in China. How they going to produce the milk? They're getting the genes, milk producing genes from the cow. Okay, they're getting the milk producing genes from the cow. They are inserting them to the bacteria. Okay, this gene is now in the bacterial DNA, and now they're making the proteins, and they are dividing very fast each 20 minutes. They are obtaining the milk from the bacteria now. This is how China is surviving in the way of products now. Okay, there are no insufficient amount of the products for such a huge population. They are using the bacteria as a help. They are inserting genes to the bacteria, and now the bacteria are doing the stuff which animals are doing. Okay, it's so super cool. So good. Any questions regarding for example, the insulin? Have you ever heard about the insulin?
Yeah.
This is very useful in the genetic research and biotechnology. For example, In the real example, I can say scientists use it to insert the genes into the bacteria, like for the insulin creations or for the insulin production or for the creating the GMOs, genetically modifying organisms. So what's the meaning of GMO? Genetically modified organism or genetically modified product. They use it, they change their genes, okay? For example, scientists use it to insert the genes into the bacteria. For example, we have in our pancreas, поджелудочная железа, we call it. In our pancreas, we have what? The cells, beta cells that produce the insulin. We are getting that genes, that DNA, and inserting to bacteria, and bacteria is producing insulin, and that's sold in our markets, insulin for the type 1 diabetes mellitus or for the type 2 diabetes mellitus. So scientists take the gene for the insulin from humans and insert it to the E. coli. The transformed E. coli now can produce the insulin for the diabetic patients. Okay, this is called what? This is called transformation. This is why the transformation is very important. Okay, good. Any questions from the transformation?
No questions.
Okay, I hope you understood. And this is, I'm sorry for writing here. Let me clear everything. So after that, transduction. What was the transduction? Can you remind me? Can someone remind me?
Yes, it is when the DNA of one bacteria travels throughout the virus to get to another bacteria.
Okay, good. So transduction, it can be generalized transduction and it can be specialized transduction. Okay, so transduction is when the virus, we know the bacteriophage carry the DNA from one bacterium to the another. Okay, it's like a delivery mistake. A virus accidentally, the virus doesn't know, it doesn't have the brain, right? In the virus, we don't have the brain. It doesn't know what it's doing. The virus is accidentally picking up some bacterial DNA and delivering it to another bacterium. There are two types of this transduction. First is called generalized. The second is called specialized. Okay, what's the meaning of generalized transduction? Meaning is random bits of any bacterial gene are accidentally packed into the virus. Accidentally packed into the virus. So let's break them down step by step. For example, we have a lytic phage. We have a lytic phage. This is the lytic phage. Lytic phage contains the information from another bacteria. Okay, it chop up the host DNA to destroy it. What's going to do? First of all, infecting the bacterium. This lytic phage is going to infect the bacterium. Okay, so it's inserting its information, but this information containing the cleavage of bacterial DNA. So it's chopping up the host DNA. This is called host DNA. This is the new bacterial DNA. So when they are inserted, they're going to be chopped into the pieces. Okay, to destroy it. We are going to destroy the bacterial DNA. Okay, when making new phages, some bacterial DNA gets mistakenly packed into the virus head. Okay, so what they're going to do, this is the information of the bacteriophage, right? This is the information of the bacteriophage. There will be the information to make the capsid. This is the capsid of the virus. This is the outer layer protection of the virus, okay? When making new phages, by the protein synthesis, they are making the new phages. We call them bacteriophages, okay? But some bacterial DNA also, the bluish one is bacterial DNA, gets mistakenly packaged into the virus head as well, into the virus, mistakenly inside of the capsid. Some bacterial DNA is inside of the capsid. So what's going to happen? These phages infect another bacterium injecting the wrong DNA. So there will be a release of the new phage from the lyse itself. When they multiply, when they increase in number, what they're going to do, they break down the cell. They're going to break down the cell. The cell is going to lyse. It's going to rupture. Then all the contents will be released out. But these phages infect another bacterium injecting the wrong DNA. This is the bacteriophage, right? But inside of the bacteriophage, we have the DNA and we have the information from previous bacteria. This is the information from the previous bacteria, okay? The bluish one here, injecting the wrong DNA. And now the new bacterium gets the foreign bacterial gene. Now this new bacteria is getting foreign bacterial gene. This is called the generalized actual packaging, okay? Imagine the virus meaning to carry the virus instructions, picks up the pizza recipe instead, okay? It delivers this recipe to another house. Now the new house bacterium can make also the pizza, okay? For example, here, imagine this virus has an information to cook a pizza, okay? Imagine, this is just imagination, okay? A virus mean to carry the virus instruction. This is actually the virus instruction. Virus instruction. Imagine this instruction is to make a pizza. Now, when it delivers to another bacteria, this bacteria also will have what? Now the new house is like a bacteria, can also make a pizza, right? So that means these genes can also make a pizza now. They have the instructions now, okay? This is how it works. This is how it all happening, this generalizing. For example, in the real life example, I can say a phage infects the salmonella. You know the salmonella?
No.
This is salmonella. It's a bacteria inside of foods. Some foods stay for a long time. When you consume them, you will get the salmonella, you will have the diarrhea, you will have the infection, GI infection. Okay, this is salmonella. Some phages, some bacteriophages infect the salmonella. They accidentally pick up a gene for the sugar metabolism and transfer it to another salmonella. Now the new salmonella can digest a new sugar it couldn't do before. So each time when the phage is getting the information from one bacteria and bringing it to another bacteria, it's bringing previous bacteria's superpower. And that new bacteria will also have that superpower. That's why we always create the new antibiotics to tackle with another bacteria. That's why we have the broad spectrum antibiotics now to tackle with those bacteria. They gaining more and more powers, okay, with three ways: with transformation, with transduction, and with conjugation. So what's specialized? Transduction? Actually, specialized meaning only specific genes near the virus's integration site are transferred. Okay, only specialized genes are transferred. This is the viral DNA. Okay, this is the bacteria. Okay, the lysogenic phage inserts its viral DNA, now inserting its viral DNA into the bacterium chromosome. This is the bacterial chromosome, right? Now they inserted that gene into the bacterial chromosome. The viral DNA stays hidden there for a while. It's staying hidden. Okay, they're staying hidden for a while. Later, it decides to excision. Later, this is hidden for a while. This is just a hidden, but after some time, they have the information to cleave, to chop, for cleaving. After some time, they are cleaving the bacterial. They're cleaving each part of the bacterial DNA because it contains the genes to cleave, okay? Later, it decides to cleave, excision. That means the excision. Okay, that means the excision. So what's going, but when it does, it accidentally takes nearby bacterial genes. When it's excising, it's accidentally taking the bacterial DNA. You see, each bacteriophage now, from the protein synthesis, they created the capsid outer layer, but each one contains the bacterial DNA now. You see, there is a viral DNA and bacterial DNA, okay? Imagine like a virus parking its car in your garage. This is your garage, and this is parking their car in your garage. When it leaves, when the virus leaves, it accidentally takes your bike. Okay, when it's leaving, it's accidentally taking up your bike. It then gives this bike to the next house to visit. You see, when they release outside, they are taking the bacterial what gene. That means a bike. Now they're going to another bacteria in inserting that or bringing this bike to another house. Okay, now this house, the new house, has both car and bike, which came from another bacteria. Okay, it's like a phage infecting the E. coli and integrating near a toxin gene. When it leaves, it takes that toxin gene and gives it to a non-toxic E. coli. Now the new E. coli is dangerous because it produced the toxin. Okay, you imagine you have a normal E. coli, which is in your digestive tract. You have a normal flora of bacteria, E. coli, which is producing vitamin K for you. Okay, you're happy together. But what's going to happen? One virus is going to come, inject, it is DNA. Now what's going to happen? This information with the virus, the toxic information is mixing with normal bacterial information, and this bacteria contains now both good portion of the DNA and bad portion of the DNA, and they are becoming now phages, and these bacteriophage is now going to another healthy bacteria in injecting this toxic ability, and now this bacteria is also toxic. From healthy E. coli, you're getting bad E. coli. Okay, this is just specific. Okay, bacteria can become more dangerous. Why this is so important, guys? Why this transduction is more important to the medicine? Because transduction can spread toxic genes, antibiotic resistance, and new metabolic traits to another bacteria. Okay, scientists use also transduction process in genetic engineering to trace the gene functions also, and also for the vaccine development, some phage therapy uses viruses to kill specific bacteria by understanding how they work. Now, what they are doing, what scientists are doing, they're injecting the normal virus. They're injecting normal virus to kill your toxic bacteria. Okay, good.
Yeah.
This is actually what's how information might show up on the test. This slide is more about the antibiotic resistance. Now you know what's antibiotic resistance, right? Imagine this is the plate where the bacteria is going to grow. Okay, this is the plate where the bacteria is going to grow. Imagine these are the peels. These are the peels. They are antibiotics. Here, for example, you have the antibiotics, which is called ampicillin. Here you have, imagine, you have a antibiotics which is called tetracycline. I call it tetracycline, okay? Tetracycline. So, and these yellow ones, what are those yellow ones? What are those yellow ones, yellow greenish structures?
Bacteria.
Mold.
Of course, they are bacteria. When I'm putting the ampicillin, this is I'm doing this is one part of the bacteria. Okay, this is one type of bacteria. This is another type of bacteria. Okay, this is another type of bacteria. Let's look. So when I'm injecting to this type of the bacteria ampicillin, they're going to die. You see those areas, they died. That means they are ampicillin sensitive. Ampicillin, they are ampicillin sensitive. When I'm injecting here, when I'm putting the tetracycline, here, the bacteria are dying with tetracycline. That means they are tetracycline sensitive, right? But if I put here, if I put this to this part, let me do like this. is another type. Okay. So when I put here the tetracycline, imagine this is tetracycline, these bacteria are not dying. Those bacteria are not dying. And we call it tetracycline resistance. I put red, I put S here. This is ampicillin sensitive. Okay. This is tetracycline resistance bacteria. Okay. So this is tetracycline sensitive. This is, imagine, ampicillin resistant bacteria. Okay. When I'm putting to these types of the bacteria ampicillin, they are not dying. But if I put tetracycline, they are dying. When I put here ampicillin, they are dying. When I put tetracycline, they are not dying. What's going on? Yes, that might happen. So after some time, those bacteria are going to grow together here. After some time, they're going to grow together. But when I put here both ampicillin, both tetracycline, they are not dying. What happened?
They're connecting
You're resistant.
to each other.
They became resistant. They did the conjugation or transformation. Which one? They did the conjugation. They did the conjugation. They used their sex pili, right? From one bacteria, another bacteria, they connected each other. They shared the plasmid, they shared the resistant gene to another bacteria. Now, both of them are resistant to both tetracycline and ampicillin. Okay? That's why when doctor prescribe you antibiotics, you have to take it in the full prescription always. Let them die fully before you get the resistance. Okay. Okay. Here it's written because of the conjugation that's happened. Please, you can read it yourself as well. Okay, good. So thank you very much. It's time to end it here. Okay, we will continue in the next lecture. Any questions to me?
I got no questions.
Okay, good. So let me take an attendance, please. Tak. We have only Mahbuba, Gulkhayo. Is Gulkhayo here? No, Saodat, Jahangir, Mahbuba. Mahbuba here. Darin is not here. Nurkhayat not here. Gulkhayo not here. Zalala not here. Saodat, Jasur. What about Munisa? Not here. Okay. Mahbuba, Saodat, Jasur. Jasur. Yeah, where is Jahangir? Jahangir, your name is not here.
Really?
Only Jasur's name, Akbar of Jasur, is here. Okay, guys. Thank you very much. Have a good evening.
Thank you too.
You're welcome.
You too. Bye.
Thank you.
Bye.
Goodbye.